right, good day. My name is Jason White. I'm a Chan Zuckerberg Initiative postdoctoral fellow at Morehouse School of Medicine in the Institute of Translational Genomic Medicine. First, I'd like to thank the organizers of this event for extending the invitation for us to participate. And second, I would like to thank those viewing this presentation for their time and consideration. Today, I'll be presenting diving into the unknown to address cancer disparities, genetic diversity, and variants of unknown significance. This section of the event is dedicated to discussing cancer health disparities, and in our lab, we believe a meaningful approach to addressing disparities lies in investigating variants of unknown significance, abbreviated VUS. Within this presentation, I will lay out the disparities that we face in cancer genomics, define variants of unknown significance, describe how variants are classified, illustrate VUS rates across populations, provide examples of variant reclassification in cancer, and highlight efforts within our research group to study VUSs in people of African ancestry. Cancer health disparities extend across multiple cancer types, or multiple disease types, and are defined as adverse differences in disease burden between different population groups. These differences include incidence, mortality, screening rates, and stage of diagnosis. The factors that contribute to these disparities are complex and interrelated and can include geography, income level, race, culture, genetics, and biological factors. Non-Europeans are significantly underrepresented in genomics databases. As you can see here in panel A, it shows the ancestral proportions of individuals within the GWAS catalog, and panel B shows the ancestral breakdown in the Genome AD database. In both examples, the number of European individuals are greatly overrepresented, making up a majority of the databases despite being a global minority. In addition, continents outside of America and Europe are poorly represented in genomics data. For example, the International Health Cohorts Consortium has far exceeded its goal to enroll 100 participants. However, American and European individuals account for 90% of enrollments. Efforts on this level require extensive and experienced collaborator networks, effective communication, solid funding, and intentional actions to target underrepresented populations. Organizations like International Health Cohorts Consortium are making strides in the right direction. However, more needs to be done to extend participant representation. Non-European Americans are also less likely to undergo genetic testing. 620,000 specimens profiled from 2013 through 2022 from Foundation Medicine were retrospectively analyzed, and they represented patients from all 50 states and showed that more than 70% of specimens were obtained from individuals of European ancestry. In 2021, the American Society of Human Genetics prepared the Human Genetics and Genomics Workforce Survey, and it showed that the majority of respondents in the genomics and genetics workforce were predominantly white. The study concluded that the genetics and genomics workforce is insufficiently diverse, lacking equitable participation of underrepresented communities based on race and ethnicity, gender, disability, and economic background, and that the workforce does not reflect the makeup of students entering the workforce pipeline, nor the domestic and global populations it serves. Now that we've walked through the disparities that we face, let's illustrate the consequences of those disparities, using prostate cancer as an example. If you conduct a general literature search, you will be able to discover several prostate cancer associated mutational patterns that, like the ones identified in this figure, such as high prevalence of temporary ERK fusion, TP53 and PTEN mutations. However, these associations were predominantly constructed using European American samples. When you do an intentional search for studies that include African Americans, what you'll observe is that the associations change in several ways, including lower rates of temporary ERK fusion and increased rates of SPINK1 and LSAM. showing the difference of intentional versus a general search. Now that we've kind of laid that groundwork, we're kind of going into the what is a variant of unknown significance, or in this case, how are variants classified in general. The most widely adopted system for sequence variant classification is the 2015 guidelines published by the American College of Medical Genetics and the Association of Molecular Pathology. Genetic variants that are uncovered during genetic testing are largely classified on a continuum from benign, likely benign, uncertain significance, likely pathogenic, and pathogenic. Benign or likely benign variants are unlikely to be associated with disease. Variants of unknown significance do not have enough evidence to be classified, and genetic variants showed to be classified as pathogenic or likely pathogenic are considered to be disease causing. And I also think it's worth noting that variants of unknown, variants that are called likely benign or benign are typically categorized that way also due to a lack of evidence, similar to variants of unknown significance. Now, how these guidelines are specifically laid out in the published document, and they were created through surveying professionals within over 100 sequencing labs and compiling empirical data. They only pertain to germline variants, and term usage differs from those used for copy number variants. Likely pathogenic and likely benign denote a greater than 90% certainty. The guidelines also recommend that clinical sequence reports should be written in clear language, identify variants using Human Genome Variation Society nomenclature, contain supporting evidence, and recommend supplemental clinical testing. Lastly, it is also recommended that all findings of pathogenic and likely pathogenic variants should be confirmed through secondary validation. The guidelines also lay out criteria that aid in determining the classification of profiled variants. As you can see here, this lays out at least a portion of the criteria for how pathogenic variants are classified. And in order for a variant to be called pathogenic, it must meet multiple criteria. The first of which meaning it must have very strong meet the very strong criteria, meaning it must be in a gene, or the variant must be in a gene where a loss of function is a known mechanism of disease. It also must meet one or more characteristics from lower evidence tiers, including strong and moderate. Population, disease-specific, and sequence databases and in silico programs can be used to aid in variant interpretation. Reference sequences, population allele frequencies, literature evidence of pathogenicity, and the protein structure impacts of a variant are all useful in accurately classifying a variant. Now, with that said, the rates of variants of unknown significance differ by ancestral groups. Non-European populations are underrepresented in research and genomic databases, as we mentioned. And so that skews genomic annotations away from identifying clinically relevant variants in these populations. This in turn leads to non-European populations having higher rates of VUSs. As in this example, it shows that African Americans have almost a 50% or 100% increase in the rate of variants of unknown significance. This lack of diversity in research, and by extension genomic databases, is an understood challenge in disparities work, as shown in these studies. And the increased rates not only apply to African Americans, but it also applies to other non-white groups such as Ashkenazi Jews, Asians, and also Hispanics. In addition, rates of variant reclassification are higher in European patients. So typically, a patient comes in, their sample is profiled, and then sometime later, those variants will be reclassified. They'll be revisited, reanalyzed, to assess any updates in known technology or evidence regarding pathogenic variants. And this is what is shown here. So a 2018 study by Slavin et al. Show that variant reclassification typically happens within 2 years of profiling, and that reclassification rates are higher in non-European patients, specifically in BRCA1 and 2 variants. By tracking nearly 1,500 variants in cancer genes originally classified originally classified as benign and reported over a 20-year period, they found an overall reclassification rate of 18.1%, with patients from non-European ancestries having higher rates of reclassification, ranging as high as 42.9% in BRCA1 and BRCA2, and 24.4% for 40 other cancer genes in patients of African ancestry than the patients from non-Hispanic European ancestry. Now, what that means is that it opens up possibilities. It opens up avenues, or the existence of these variants of unknown significance opens up possibilities and opportunities for lines of research. One database or one new project that has come out of the NIH is a research program called All of Us. And within this platform and this project, there have been 1 billion genetic variants identified, including more than 275 million that were previously unreported, more than 3.9 million of which had coding consequences. So the presence of these variants of unknown significance, or the issue is that once you identify them, then you can take them and characterize them and see if they in fact are pathogenic, disease causing, whether they're related to disease development, progression, or severity. Within All of Us platform, there continues to show the same pattern that we showed before, where non-European like individuals have increased rates of variants of unknown significance. And as you can see here in this figure, that is a consistent pattern across multiple areas of research, where there's cardiology, endocrinology, oncology, nephrology, even in screening patterns or screening projects. There continues to be that increase. So again, all of these variants of unknown significance provide a well of leads as far as addressing the disparities that that have been discussed in this presentation and in others. As far as our contributions to this effort, we have published multiple manuscripts focused on people of African ancestry within breast cancer and prostate cancer that include not only American cohorts, but also cohorts in Northern Africa. And what we've been able to identify are specific variant patterns that are associated with African ancestry. And so we've identified specifically variants in related to DNA damage repair that we believe are associated with African ancestry, but also that can contribute to tumor development, including the BRCA1 variant RS79917, which has been shown to impair DNA damage repair, thus enabling tumors to form. But we also have other leads across in BRCA1, BARD1, and also BRCA2. Now, with all of that said, this is only one avenue for addressing the disparities that we've talked about. We not only in order to address the disparity, you have to integrate data and information from multiple domains, not only genomics, but also exposomics, meaning a person's environment, also access and social determinants of health. And taking all this information in together is the only way that ultimately we're going to be able to address the disparities in a comprehensive and long-standing way. All right, to that end, in an effort to integrate data from multiple domains, our group was successful in competing for a funding opportunity funded by the Cancer Research UK and also by the National Cancer Institute, called the Cancer Grand Challenge. Our team SAMBI, which stands for Social, Ancestry, Molecular, and Biological Analysis of Inequalities, we're charged with the challenge to understand the mechanisms through which genetics, biology, and social determinants affect cancer risk and outcomes in diverse populations to motivate interventions to reduce cancer inequities. In this challenge, what we seek to do is to generate functional and mechanistic insights into cancer inequities by generating new transdisciplinary approaches applied to diverse populations. Now, what that means is that we're endeavoring to collect 40,000 samples across multiple continents, which would include Africa, the UK, and America, run those samples through multiple omics platforms in an effort to bring together enough information to fully understand and characterize pancreatic, prostate, and breast cancer in people of African ancestry. Now, the beautiful thing about this project is it will build a framework that can be applied to any minority group or any group really at all, any specific disease cohort that one would want to study. And so our hope is that it would just be the beginning of a new approach that changes the focus or in a way that allows underserved groups to fully be able to harness the power of precision medicine and to be and to not be harmed or not to perpetuate the disparities that we've discussed. With that said, I thank you so much for your time and listening. If you need me, I can be reached at jawhite@msm.edu. Again, thank you for your time, and I appreciate you.